So this is our sample, and we are extracting the DNA from banana. The best sample that can be used for DNA extraction is strawberries because it has got lengthier and much more quantity of DNA. So the samples, but then strawberry is available only for few seasons, and that is why the next best alternative is banana. But the problem that we get when we take banana is there is a lot of fiber that needs to be cleaned up. So this is a mortar and a pestle. What I'm going to do is I'm going to just smash the banana. So I've just taken it and I'm just, it's slippery also. We'll just somehow mash it a little bit. Okay, now once we have mashed up the banana, what we are going to do is we'll add detergent solution. So what this detergent solution is going to do is it is going to dissolve the cell wall, the pectin, the cellulose, and it is going to basically crack open the plant cell. So I'm just adding this detergent. Now once you add this detergent, care should be taken that we need to avoid froth formation. Otherwise it is going to interfere with the extraction of DNA. So be gentle and just incubate this sample for around 5 minutes. Allow the cell walls to open up and gentle mashing is to be carried out. Okay, so we have mashed the banana into a pulpy mass. Now we are going to add sodium chloride. Now what sodium chloride does is it has got sodium ions which are positive and chlorine ions which are negative, and you know that DNA is negatively charged molecule because it has got a phosphate background. So sodium basically is going to neutralize and mask the negative charges of the DNA, so that when we go for ethanol precipitation of DNA can now leave water and go to the alcoholic phase. Otherwise the hydrogen bonding with water will not allow the DNA to leave the aqueous phase. So just to mask the negative charges, we are adding NaCl. Other than that, NaCl is also going to upset the osmotic balance of the cell, and it is also going to inactivate the enzymes, because we know that enzymes function in an optimal level of pH and salt concentration. So we are also trying to stop some of the enzymes like DNase, because if DNase are not deactivated, they will destroy our DNA. So we have added NaCl. Now what we are going to do is we need to mash it through a cheesecloth just to reduce the amount of fiber. Okay, so we are just passing it through a cheesecloth. So we are just going to mash it up. Okay, so we have got our sample. Now what we are going to do is we'll transfer this sample to a test tube. Okay, so we have got our sample, and now what we are going to do is we'll take chilled ethanol and we will slowly add from the sides. And as you can see, there is a cloggy mass or a foamy mass which is being formed at the interface of ethanol and the sample, and the DNA is present just at that interface. So if we keep it for some time, we'll be able to observe this data. So if you can see, this layer of white color cobweb structure is the DNA. It's a very crude form of DNA. Okay, thus our DNA crude extraction concludes.